This is a tardigrade. It's about half a millimeter long. It lives in puddles, in soil, in the moss on your garden wall. It's so common, you've almost certainly walked past millions of them today without noticing. Scientists call it a water bear, which is the most aggressively endearing name they could have given something that looks like a microscopic warthog. Now here's what you need to know about this ridiculous little creature. In 2007, the European Space Agency strapped a container of tardigrades to the outside of a satellite and launched them into orbit. No suit, no ship, no oxygen. Just a box of water bears bolted to the hull of a spacecraft directly exposed to the vacuum of space. They floated there for 12 days. No air, no pressure, radiation pouring into them from every direction. Temperature swings between -272 and +150 degrees Celsius. The full, unfiltered fury of deep space. You know what happened when they brought the tardigrades back? Most of them were fine. Some of them were even reproducing. They had survived conditions that would vaporize, freeze, irradiate, and otherwise obliterate virtually every other living thing on Earth. These microscopic, eight-legged, arguably adorable little monsters had just passed the first qualification test for interplanetary travel. And here is where it starts getting uncomfortable. Because if life can survive the vacuum of space, if even a tiny fraction of organisms can endure the brutal conditions between worlds, then an idea that scientists have debated for over a century starts to look less like speculation and more like the most disturbing possibility in the history of biology. What if life didn't start on Earth? What if it arrived here? What if instead of Earth being the birthplace of life, it was simply the landing pad? What if every living thing that has ever existed on this planet, every bacterium, every dinosaur, every human being watching this video, is the descendant of something that fell from the sky? This is Panspermia, and by the end of this documentary, you'll either believe it's the most elegant explanation for the origin of life ever conceived, or you'll believe it's the most terrifying. Or both. Probably both.
Let's start with a problem. A very specific, very stubborn, very inconvenient problem. And it's this. Life on Earth appeared almost immediately after Earth stopped being a hellscape. The Earth formed about 4.54 billion years ago. For the first several hundred million years, the planet was a nightmare. The surface was molten. Asteroids were bombarding it constantly in what geologists call the Late Heavy Bombardment, a period of sustained cosmic artillery fire that would have sterilized any life that dared to exist. This epoch ended roughly 3.9 billion years ago. The oldest confirmed evidence of life on Earth dates to approximately 3.5 to 3.7 billion years ago. Some controversial evidence pushes that back even further, to 4.1 billion years ago. So, after the bombardment ended, life showed up almost immediately, give or take a few 100 million years. A few 100 million years sounds like a long time to you because you were a mammal with a 40-year average attention span. But in geological terms, a few 100 million years is essentially overnight. It is not enough time, not even close. Scientists have been trying to figure out how life started on Earth for decades. The Miller-Urey experiment in 1952 famously showed that you can create amino acids, the building blocks of proteins, by zapping a mixture of simple gases with electricity, simulating lightning in a primordial atmosphere. Great. Wonderful. We can make amino acids. Here's the problem. Amino acids are not life. They are to life what individual letters are to the complete works of Shakespeare. You've got the letters. Congratulations. Now explain how you get from a scattered pile of letters to Hamlet, spontaneously, in a few 100 million years. A living cell is almost incomprehensibly complex. You need a self-replicating molecule, DNA or RNA, that can store and transmit information. You need proteins that can carry out chemical reactions. You need a membrane to contain everything. And you need all of these components to appear at the same time in the same place and begin working together with enough coherence to actually replicate. The mathematician Fred Hoyle, the same man who coined the phrase Big Bang as a term of mockery, calculated the odds of the simplest functional protein assembling itself by chance at roughly 1 in 10 to the power of 40,000. For reference, they're estimated to be 10 to the power of 80 atoms in the observable universe. The number of possible protein configurations is so vast that random chemistry simply doesn't have enough time or enough atoms to find the right one. Hoyle described this as equivalent to a tornado sweeping through a junkyard and assembling a Boeing 747. His analogy was designed to be devastating, and statistically speaking, he had a point. So the timeline is wrong. Life appeared too fast. The chemical odds are staggering. Earth's window between no longer being bombarded by asteroids and teeming with microbial life is criminally short by the standards of what abiogenesis, life arising from non-life, is supposed to require. This is where panspermia enters the conversation, not as a handwave or an easy out, but as a genuine scientific solution to a genuine scientific problem. What if you don't need to explain how life started on Earth in a few 100 million years? What if life started somewhere else, with billions more years to develop, and then arrived here, pre-assembled? The universe is 13.8 billion years old. Earth is only 4.54 billion years old. That means there are stars, and presumably planets, that had 9 billion years of head start on us. 9 billion years is enough time to evolve life, develop microbial complexity, and scatter that life across the galaxy many times over. Panspermia doesn't answer the ultimate question of where life came from originally. It just says that the starting point wasn't here. It moves the problem, yes, but it also takes enormous pressure off the timeline because the originating world had incomparably more time to work with. There are 3 main flavors of panspermia: lithopanspermia, life traveling inside rocks knocked off planets by asteroid impacts; radiopanspermia, life traveling on the pressure of light itself, like microscopic spores blown by the cosmic wind; and directed panspermia, the genuinely unsettling possibility that someone put life here on purpose. You might think directed panspermia is the fringe territory of conspiracy theorists. It's not. In 1973, Francis Crick, Nobel Prize winner, co-discoverer of the structure of DNA, one of the most important scientists of the 20th century, co-authored a paper proposing that life on Earth may have been deliberately seeded by an extraterrestrial civilization. Crick wasn't a crank. He was responding to exactly the same statistical problem we just discussed. His reasoning was elegant and disturbing. The genetic code, the specific system by which DNA triplets encode amino acids, is essentially universal across all life on Earth. Every bacterium, every fern, every blue whale uses the same code. There are about 10 to the power of 70 possible genetic codes that could work equally well. We use one, just one, the same one, everywhere on Earth, without exception. That doesn't prove panspermia, but it does scream single origin. All life on Earth appears to descend from one common ancestor, one founding population. The question is where that founding population came from, and the answer might be written in the rocks.
Here is how lithopanspermia works, and why it's more plausible than it sounds. When a large asteroid or comet hits a planet, the impact energy is enormous. The surface rock near the impact point is subjected to pressures so intense that it can be accelerated to escape velocity, fast enough to leave the planet entirely before being vaporized. This sounds counterintuitive. You'd expect a rock to be vaporized by an impact that powerful, but the physics works out. Material on the periphery of the impact zone, far enough from the center to avoid direct vaporization, gets hit by the pressure wave and can be flung into space as intact fragments. We know this because we've found them on Earth. There are roughly 270 meteorites on Earth that we've confirmed came from Mars. We know they're from Mars because the chemical composition of gas bubbles trapped inside them matches exactly the Martian atmosphere as measured by the Viking landers. These rocks were blasted off the Martian surface by ancient impacts, drifted through space for millions of years, and eventually landed here. The journey from Mars to Earth takes anywhere from a few thousand years to a few million years, depending on the orbital dynamics. Now, here's the question that matters. Can life survive that journey? The conditions in deep space are brutal. Temperature near absolute zero, intense cosmic radiation, complete vacuum, duration of millions of years. It seems impossible that anything biological could survive. But then you remember our friend the tardigrade, and it's not alone. Bacterial spores, particularly those of the genus Bacillus, are among the most radiation-resistant organisms on Earth. They can survive doses of ionizing radiation hundreds of times higher than would kill a human. They can remain dormant, metabolically inert, for millions of years. Scientists have revived bacterial spores from 250-million-year-old salt crystals. That's older than the dinosaurs. Dormant for 250 million years and still viable. Now, put those spores inside a rock. A meter of solid rock provides extraordinary shielding against cosmic radiation. The interior of a large boulder drifting through space could maintain a relatively protected environment for biological material for timescales measured in millions of years. Studies have shown that bacterial spores embedded in granite can survive radiation doses equivalent to traveling through space for up to 25 million years without full sterilization. 25 million years! Mars and Earth are close enough that transfer happens in thousands to millions of years. The math, improbably, works. So the transfer scenario looks like this. A large impactor hits Mars 4 billion years ago, when Mars still had liquid water and a thicker atmosphere and, according to a growing body of evidence, possibly microbial life. The impact ejects rocky debris into space. Some of that debris contains organisms protected inside the rock matrix. The organisms enter a dormant state. The rock drifts through the inner solar system. Over thousands to millions of years, the orbit intersects with Earth. The rock enters Earth's atmosphere. Atmospheric entry is violent. The exterior of the meteorite heats to thousands of degrees, but the interior stays cool. The thermal pulse from entry lasts only seconds to minutes. The inside of a meter-scale meteorite barely warms at all. The organisms inside experience less heating than a pizza in a microwave. The meteorite hits a primordial ocean. It dissolves over millennia. The organisms, still viable after their impossible journey, encounter liquid water and simple chemistry. They do what life does. They survive, they replicate, they colonize, they evolve. 4 billion years later, their descendants are watching a YouTube documentary about them. This is not science fiction. Every step of this process has been independently verified. We know large impactors eject surface material. We know Mars rocks reach Earth. We know spores can survive in rock matrices for millions of years. We know atmospheric entry doesn't kill interior material. Each individual domino has been confirmed. We've never watched the whole chain fall at once, but every piece is real. And here's the thing about Mars that nobody talks about enough. Mars had liquid water before Earth had life. Mars had a magnetic field, a thick atmosphere, and stable surface oceans roughly 4.1 to 3.8 billion years ago. That's exactly when Earth was being hammered by the late heavy bombardment. Earth was a sterilizing hellscape, while Mars was hospitable. If life arose on Mars first, which is entirely plausible given Mars's earlier habitability, then the Panspermia chain from Mars to Earth becomes not just possible, but almost expected. We were neighbors. We were exchanging rocks constantly. If one house had a fire, the other house was going to catch some sparks. And it works both ways. Earth to Mars transfer also happens. We've found Earth rocks on the Moon. Rocky debris from our world has been scattered throughout the inner solar system for billions of years. This is the central, mind-altering implication of lithopanspermia. The planets of the inner solar system may not be separate, independent biological experiments. They may be one system, a single biosphere spread across multiple worlds, constantly exchanging genetic material, constantly seeding and reseeding each other, more like organs in a body than isolated islands in a void. And if it works within our solar system, why would it stop here? In the early solar system, stellar densities were higher, stars were closer together. The rate of cometary exchange between solar systems may have been significant. Studies suggest that billions of rocks were exchanged between solar systems during the formation phase of the galaxy. The Milky Way, at its densest stellar cradle, was a biological mixing bowl. Life, once started, might propagate through an entire galaxy the way a cold spreads through an office building, slowly, messily, without anyone's permission, without any intelligence required. Just rocks and time and the unrelenting indifference of orbital mechanics. But wait, before we get to what this means for the galaxy, let's talk about the evidence. Because might and possibly are fine for a philosophy seminar. Science demands proof. And the evidence, as it turns out, is considerably more interesting than most people realize.
On the morning of September 28, 1969, residents near Murchison, Victoria, Australia, heard a series of explosions in the sky. A large meteorite had broken apart, entering the atmosphere, scattering fragments over a wide area. Within days, over 100 kilograms of material had been collected. This would turn out to be one of the most scientifically valuable rocks ever studied. The Murchison meteorite is a carbonaceous chondrite, a type of ancient organic-rich space rock dating to the very earliest period of the solar system, roughly 4.6 billion years old. It formed before Earth did. It is older than our planet. When scientists analyzed the meteorite, they found amino acids, not just any amino acids, over 90 different types, including some never found in nature on Earth. Some of them were the same amino acids used in earthly biology: glycine, alanine, glutamic acid, the same letters our DNA is written in. Here's the critical detail. Amino acids can exist in two mirror image forms, left-handed and right-handed, like your two hands. Chemistry produces them in equal amounts, but all life on Earth uses only left-handed amino acids. Every protein in every living thing on this planet is made of left-handed amino acids exclusively. If you contaminated the Murchison meteorite with Earth biology, it would skew heavily left-handed. The meteorite shows both forms, with only a slight left-handed excess. That slight excess is consistent with theories about how polarized life in star-forming regions could preferentially produce left-handed molecules. Translation: these amino acids are not contamination from Earth. They formed in space, before our solar system existed, in the molecular cloud that eventually collapsed to form our sun and planets. The raw ingredients of life were raining down on the early Earth from space, not metaphorically, literally. But the Murchison meteorite, as revelatory as it was, had nothing on what came next. On August 7, 1996, President Bill Clinton stood outside the White House and made an announcement that nearly stopped the world. A Martian meteorite, catalogued as ALH 48001, had been found to contain what NASA scientists believed were signs of ancient microbial life from Mars. Possible fossilized bacteria, organic compounds, mineral structures consistent with biological processes on Mars. Life on Mars. The world lost its collective mind. The meteorite had been found in Antarctica in 1984, hence ALH 48001, but its Martian origin wasn't confirmed until later. It was estimated to have landed on Earth 13,000 years ago after spending 16 million years in space, after being blasted off Mars roughly 3.9 billion years ago. When it left Mars, Mars still had liquid water. Inside ALH 48001, scientists found polycyclic aromatic hydrocarbons, organic molecules associated with biological decomposition. They found magnetite crystals with unusual morphology similar to those produced by certain Earth bacteria. And they found those infamous worm-like structures, tubular forms 20 to 100 nanometers across, resembling fossilized bacteria. The scientific community erupted. Critics immediately pointed out that the putative bacteria were far smaller than any known Earth microbe, so small that fitting all the molecular machinery of life inside them would be physically impossible. Others noted that the organic compounds could have been terrestrial contamination. Others questioned the magnetite formation process. The debate continues to this day. But here is something important. The skeptics never definitively disproved the biosignature hypothesis. They raised doubts, they offered alternatives. What they did not do is explain away all of the observations simultaneously. The best current assessment is probably not life, but not proven otherwise. That ambiguity is doing a lot of work. Move forward now to some of the most exciting planetary science of the past 2 decades. Saturn's moon Enceladus is a small, icy body roughly 500 kilometers across. It should be frozen solid and completely geologically dead. It is not. In 2005, the Cassini spacecraft discovered enormous plumes of water vapor erupting from Enceladus's south pole, geysers shooting material 800 kilometers into space. Cassini flew through those plumes and tasted them. The findings were extraordinary. The plumes contain water vapor, molecular hydrogen, carbon dioxide, methane, and complex organic molecules, including some with masses over 200 atomic units, suggesting large, ring-bearing organic structures that on Earth would be associated with biological chemistry. Molecular hydrogen specifically is significant because it's produced when water reacts with rock at high temperature, a process called serpentinization, which on Earth is a major energy source for deep-sea microbial communities.
Hydrothermal vents on Earth's ocean floor support entire ecosystems independent of sunlight. No photosynthesis, just chemistry. The reaction of hot water with rock, producing chemical gradients that microbes harvest for energy. Life without sunlight. Life fueled by geology. Life that could exist anywhere in the universe where you have liquid water and rocky seafloor. Enceladus has all of that. A liquid water ocean beneath the ice, in direct contact with a rocky core with hydrothermal activity. It is, by every metric we understand, potentially habitable. And it's venting its ocean contents directly into space, where we can analyze it without even landing. Then there's Europa, Jupiter's moon. Similar story, liquid water ocean beneath an ice shell, potentially twice the volume of all Earth's oceans. No confirmed volcanic activity, but tidal heating from Jupiter's gravity. Possibly habitable. Possibly teeming with something. Titan, Saturn's largest moon, a thick nitrogen atmosphere, thicker than Earth's, lakes of liquid methane, organic chemistry happening on a planetary scale. Not habitable in the way we understand it, but a giant natural laboratory for prebiotic chemistry of a completely alien kind. And then, most unexpectedly, Venus. In September 2020, a team of astronomers announced the detection of phosphine in Venus's upper atmosphere. Phosphine, pH3, is a molecule that, on rocky planets, has only 2 known sources: industrial production by humans or biological processes. On Earth, it's produced by anaerobic bacteria. Nobody had a good non-biological explanation for how it got into Venus's clouds. The reaction was immediate and chaotic. Some scientists called it a potential biosignature, evidence of microbial life floating in Venus's temperate cloud layer, where temperatures are around 30 degrees Celsius and conditions are, in theory, survivable. Others challenged the detection methodology, and follow-up analyses debated the concentration levels. The debate is unresolved, but here's the pattern emerging across our solar system: water on Mars, organics on Murchison, possible biology in ALH 84001, hydrothermal vents on Enceladus, habitable oceans on Europa, complex chemistry on Titan, potential biosignatures on Venus. The solar system is absolutely littered with the conditions required for life. And if life can move between worlds via panspermia, then a single origin event could explain all of it. Not life evolving independently everywhere, but life starting once and spreading. The entire inner solar system as one interconnected experiment. Now here's where it gets genuinely interesting and genuinely uncomfortable. Because if panspermia is real, there's one implication that most science communication carefully avoids. If life spreads through the galaxy like an infection, and the evidence increasingly suggests it can, then the galaxy isn't empty. The galaxy is alive, full of bacteria, full of microbial ecosystems thriving around stars we've never visited, full of life that has been there for billions of years. And yet, and this is the part that keeps astrobiologists lying awake at night, we appear to be alone. We have not detected a single signal from a single intelligent civilization. The galaxy is full of life and full of silence simultaneously, which means the silence isn't about life, it's about intelligence, which means the great filter, the thing that stops simple life from becoming complex life, from becoming intelligent life, from becoming civilizations, is somewhere between bacteria and us. Still ahead, still waiting. We'll get to that. But first, we need to talk about a problem we've already created, a contamination nightmare we may have set in motion, quite literally, decades ago.
NASA has a department called the Office of Planetary Protection. Their job is to ensure that we don't contaminate other worlds with Earth biology, and that we don't bring unknown biology back to Earth from other worlds. They operate under international treaties. They employ microbiologists, engineers, and lawyers. They take their job extremely seriously. The Perseverance rover, NASA's most advanced Mars lander to date, launched in 2020, was assembled in one of the cleanest rooms on the planet. Every component was heat sterilized, irradiated, and rigorously cleaned. A team of scientists spent years ensuring the spacecraft was as biologically clean as humanly possible. The build process cost approximately $2.7 billion. Thousands of hours went into planetary protection protocols. Every surface was swabbed. Every component was cultured. Every procedure was documented. Then a technician sneezed near it. Oops. This is not a joke. This is a documented problem. Research published in the journal Microbiome found that NASA's clean rooms, despite all protocols, harbor hundreds of species of bacteria. These aren't random contaminants. They're specifically the species most capable of surviving extreme conditions, the most radiation resistant, the most desiccation tolerant, the most heat resistant. In other words, by trying to clean spacecraft, NASA created an inadvertent selection pressure for the most extremophile bacteria on Earth. The cleaning process killed the weak ones and let the toughest ones survive. You couldn't design a better experiment for selecting the exact organisms most likely to survive a trip to Mars if you tried. A 2014 study estimated that the Curiosity rover, despite its cleanroom processing, likely carried at least 65 known bacterial species to Mars. Not confirmed, this is an estimate based on DNA sampling of the spacecraft before launch. But the number is not zero. The number has never been zero. Somewhere on the Martian surface right now, there are Earth bacteria. Dead, most likely. The Martian surface is bombarded by ultraviolet radiation at levels that would kill most organisms in seconds, but not all. The Martian subsurface, protected by meters of rock from the radiation, is a different matter entirely.
If any Earth bacteria hitchhiked on a lander and were deposited near a place where liquid brine exists in the Martian subsurface, which recent radar data suggests actually exists, then the contamination problem becomes real. Earth biology established on Mars before we know whether Mars biology exists. The most expensive, most consequential biological mistake in human history. If that happens, we may never be able to determine whether life we find on Mars is genuinely Martian or whether it's an Earth expatriate we accidentally exported. The entire scientific question of independent life origination on Mars could be permanently poisoned by our own sloppy arrival. We would have answered one of humanity's greatest questions by ruining it. Now let's flip the contamination problem around. Because we're not just sending things to Mars, we're planning to bring things back. The Mars Sample Return Mission, a collaboration between NASA and the European Space Agency, plans to retrieve rock samples collected by the Perseverance rover and bring them back to Earth. This is scientifically essential. We cannot do the kind of analysis required to detect biosignatures without Earth-based laboratory equipment. You can't send an electron microscope to Mars, you have to bring the rocks home. The samples are scheduled to return in the early 2030s. They will be contained in sealed tubes, enclosed in a capsule, and transported to a dedicated Biosafety Level 4 facility, the highest level of biocontainment, equivalent to facilities that handle Ebola and other lethal pathogens, for analysis.
The assumption underlying all of this is that Mars, being apparently hostile and lifeless by surface standards, probably doesn't harbor anything dangerous. The word probably is doing tremendous heavy lifting in that sentence. If Mars harbors life in its subsurface, which we cannot rule out, then we have no idea what that life is, what it's capable of, whether Earth organisms have any defense against it, or whether our biosafety protocols are sufficient to contain something that evolved on a completely different world under completely different evolutionary pressures. We spend significant resources on asteroid defense because the probability of a major impact in any given century is perhaps 1 in 10,000. The probability that Mars samples contain viable biology is unknown. It could be 0, but it could also be much higher than 1 in 10,000. And the consequences of getting it wrong are not a local catastrophe. They're a planetary one. Here's the irony that nobody seems to appreciate. If panspermia is real, this contamination problem may be pointless. If life already travels between worlds routinely, if Mars rocks have been landing on Earth for billions of years, and Earth rocks have been landing on Mars, then the two biospheres have already been exchanging biological material for eons. Any Martian biology that could establish itself on Earth has already had billions of years of chances to do so via natural meteorite transfer. We may already be contaminated with Mars biology. Mars may already be contaminated with ours. The panspermia transfer may have been ongoing for so long that worrying about a sample return capsule is like closing the barn door after the livestock has been circulating freely between continents for 3 billion years. Or panspermia might not be real, and Mars might harbor biology that has never touched Earth, and we're about to bring it home in a sealed canister, and our planetary protection protocols represent the most important $3 billion humanity will ever spend. Either way, we're in an impossible position. If panspermia is real, the contamination protocols are probably unnecessary. If panspermia is not real, the contamination protocols may be the only thing standing between us and an extinction-scale biological event. We genuinely don't know which world we live in. And there's one more piece of evidence that makes the contamination question even more complicated. In 2013, a team of British scientists launched a balloon to 27 kilometers altitude, well above the stratosphere, far above any weather systems that could carry terrestrial biology upward, and collected atmospheric samples. They found diatoms, fossilized microorganisms, intact, floating in the stratosphere at altitudes they could not have reached by any known upward biological mechanism. The team concluded, controversially, that the organisms had entered Earth's atmosphere from space. The mainstream scientific community was skeptical. Contamination during collection was suggested, the methodology was challenged, but the samples were real.
The altitude was confirmed, and nobody provided a completely satisfying terrestrial explanation for how intact fossil diatoms ended up 27 kilometers above the surface. The upper atmosphere, it turns out, is a complex, poorly understood environment that we've only recently begun to sample systematically. What's up there, whether it's raining down from space or floating up from Earth, we are only beginning to understand. And that brings us to the conclusion that the entire preceding 48 minutes of this documentary has been quietly building toward, the one that ties together the tardigrades and the Murchison meteorite, and the ALH 84001 controversy, and the Enceladus plumes, and the contamination nightmare. If panspermia is real, and the evidence suggests it works at least locally within solar systems, and if the galaxy has been exchanging rocky material for billions of years, then life is not rare. Life is the default state of any world with liquid water and chemistry. Life is everywhere. Simple life, microbial life, bacterial life, everywhere. And the galaxy is still silent.
Let's talk about what panspermia, if real, means for the Fermi paradox, because the implications are not comforting, not even slightly. The Fermi paradox, the observation that the universe should be teeming with intelligent life but appears completely silent, has dozens of proposed solutions. Maybe the origin of life is vanishingly rare. Maybe intelligence is a fluke. Maybe civilizations destroy themselves before they become spacefaring. Now, add panspermia. If life spreads between worlds routinely, then the probability of life arising is not defined by local chemistry on a single planet. It's defined by the cumulative chemistry of an entire planetary system, or, at galactic scale, by the entire biosphere of a star cluster. Life becomes easy. Life becomes almost inevitable wherever liquid water and organic chemistry exist. The Drake equation has a term called F sub L, the fraction of planets where life actually arises. Without panspermia, optimistic estimates put this at maybe 0.01 to 0.1. With panspermia operating galaxy-wide, F sub L could be close to 1.0 on any world with liquid water. There are estimated to be roughly 11 billion potentially habitable Earth-sized planets in the Milky Way. Factor in moons with subsurface oceans, Enceladus-type worlds, and the number is far higher. With panspermia, potentially billions of these worlds carry life. Billions of inhabited worlds, and we've detected none of them. Because, and this is the knife in the stomach, simple life doesn't make noise. Bacteria don't build radio transmitters. Archaea don't launch satellites. Extremophiles don't vote, war, terraform, or develop philosophies about their own loneliness. This is the Great Filter. The concept, developed by economist Robin Hanson, posits that somewhere between the origin of the universe and a spacefaring, galaxy-colonizing civilization, there is a filter, a barrier that almost no species crosses. The question that matters existentially is, where is the filter? Scenario 1. The filter is behind us. The hard step was the origin of life, or the development of the eukaryotic cell, or the emergence of multicellular organisms, or the development of intelligence. One of those transitions is so improbable that almost nothing survives it, and we got lucky. We crossed the filter. It's behind us. If that's true, the future is potentially wonderful. We're a rare exception in a mostly empty universe. Space is ours to explore. Scenario 2. The filter is ahead of us. The origin of life is easy. Panspermia made sure of that. Complexity develops routinely. Intelligence emerges with some regularity. But somewhere between intelligence and lasting interstellar civilization, something kills everything, every time, without exception. Maybe it's nuclear war. Maybe it's runaway artificial intelligence. Maybe it's climate catastrophe. Maybe it's the relativistic kill vehicles that another documentary on this channel has extensively detailed, civilizations destroying each other the moment they become visible. Here's the terrible logic. If panspermia is real and life is everywhere, then the Great Filter is almost certainly not the origin of life. Life survives. Life spreads. Life is easy.
That means the filter, whatever kills civilizations before they become interstellar, is at some later stage, at a stage we haven't reached yet. We are a species that evolved intelligence roughly 300,000 years ago. We developed radio transmission 130 years ago. We entered space 65 years ago. We have not yet experienced whatever it is that kills civilizations. We have not crossed whatever filter awaits us. Panspermia makes the universe feel full, alive, interconnected, and then hands you the implication that the universe is also a graveyard of intelligent species who didn't make it, surrounded by bacterial life that thrived for billions of years on the ashes of civilizations that nearly reached the stars. This is the loneliest version of a full universe imaginable. Life is everywhere. Intelligence is nowhere. Or, if it existed, it's gone. And we are here, in our brief and fragile window of technological adolescence, broadcasting our existence, contaminating our neighbors, planning to bring alien samples home, debating whether to try to talk to the void, blissfully, terrifyingly unaware of whether the filter is behind us or directly in our path. But here is what makes this era genuinely extraordinary. Within the next 2 to 3 decades, we will likely know.
The James Webb Space Telescope is already capable of analyzing the atmospheric chemistry of exoplanets. If we detect oxygen, methane, and carbon dioxide together in an exoplanet's atmosphere, combinations that shouldn't coexist without biological replenishment, we'll have strong evidence of life around another star. The Europa Clipper, launching in 2024, will conduct detailed analysis of Europa's subsurface ocean. If we find biology there, on a moon of Jupiter, in a world that shares an evolutionary heritage with Earth via Panspermia, then we will have confirmed that life spreads between worlds and that the solar system is a single biosphere. And then, Mars sample return will bring us 30 grams of ancient Martian rock. Inside those 30 grams might be the answer to the most important question humanity has ever asked. Did life arise independently on another world, or did it arrive from ours? Or, and this is the option that changes everything, is it genetically related to us? If Martian life shares the same genetic code as Earth life, the same left-handed amino acids, the same DNA to protein translation system, then panspermia is confirmed beyond reasonable doubt. Life on Earth and Mars descended from a common ancestor. We are biological cousins. The two worlds share a family tree. If Martian life is completely alien, different chemistry, different code, different architecture, then life arose twice, independently, in the same solar system, suggesting that life is so easy to create that it's essentially inevitable anywhere conditions are suitable. Either result is civilization-altering. Either result answers the question that has haunted biology since Darwin. Either result is the most significant scientific discovery in human history. Either life is so hardy and ubiquitous that it spreads through the galaxy like seeds in the wind, or life is so inevitable that it doesn't need to spread. It just keeps happening again and again, wherever the conditions are right. Both possibilities lead to the same conclusion. We are not special. We are not alone in the biological sense. We are part of something incomprehensibly larger than we imagined. The galaxy might be an ancient, enormous, interconnected living system, billions of years old, carrying life from world to world in the bellies of meteors, the way ocean currents carry seeds between continents. Every impact an accidental planting, every volcanic outburst an accidental sterilization, every habitable world a garden tended by no one, proliferating according to no plan. And somewhere in that garden, one species of upright primates on one small world around one unremarkable star became aware of it, began to probe it, began to ask whether they were part of it, began very carefully and not entirely successfully to avoid contaminating the experiment. We are made of star stuff, as Carl Sagan famously said. We are also, it seems increasingly likely, made of space stuff. cosmic travelers, children of the interplanetary highway, living descendants of organisms that may have crossed the void between worlds and found this blue, water-drenched rock and decided, without deciding anything at all, to stay. And so did that ridiculous, endearing, unkillable little water bear, who has survived everything the universe could throw at it, who has outlasted ice ages and extinctions and mass bombardments and direct exposure to the vacuum of space, who has been here, in some form, since long before we had words for any of this. It was here before us. It will be here after us, and if panspermia is real, something very much like it was probably here before Earth was Earth. We asked where life came from. The answer might be everywhere, always, in spite of everything. The question worth losing sleep over isn't whether life spread to Earth, it's whether intelligence, ours, will survive long enough to find out.